The natural world harbors organisms with extraordinary abilities. These capabilities often challenge our conventional understanding of life. They redefine concepts such as perception and recovery in biological systems. This exploration will focus on three such remarkable creatures. Each organism exhibits unique biological phenomena. Their existence pushes the boundaries of known biological limits. Certain species exhibit unique biological mechanisms that challenge typical life cycles. These mechanisms allow them to achieve extraordinary forms of longevity. Such organisms often possess specialized cellular or genetic adaptations. These adaptations enable them to repair damage, resist aging, or even reverse their life stages. Studying these species provides insights into the potential for defying common biological limits. The Turritopsis dohrnii, commonly known as the immortal jellyfish, is a unique marine species. It possesses an extraordinary biological capability to revert to an earlier developmental stage. This process allows the adult jellyfish to transform back into its juvenile polyp form. This remarkable transdifferentiation mechanism grants the species potential biological immortality. Jellyfish exhibit a unique biological process known as transdifferentiation. This process is initiated in response to various environmental stressors. It also occurs following physical injury or the onset of senescence. Transdifferentiation allows the jellyfish to revert its cellular state. Cell dedifferentiation is a biological process where mature cells revert to a less specialized state. This transformation involves cells losing their specific characteristics and functions. The cells effectively become younger and undifferentiated, similar to stem cells. This process can lead to the organism returning to an earlier developmental stage, such as a polyp. Jellyfish possess a unique biological mechanism allowing them to restart their life cycle. Instead of succumbing to death after reproduction or aging, they can revert to an earlier life stage. This process enables them to bypass the typical biological endpoint of mortality. Consequently, certain jellyfish species are considered theoretically biologically immortal. Biological immortality refers to a state where an organism does not experience an increase in mortality rate with age. This mechanism allows the organism to bypass the typical aging process, defying senescence. It presents a unique case of perpetual rejuvenation, maintaining physiological function indefinitely. Such organisms effectively escape the natural limits of lifespan observed in most species. Biological immortality refers to organisms that do not age or have a negligible rate of aging. However, this state does not imply invulnerability to all forms of harm or death. Such organisms remain susceptible to predation from other species within their ecosystem. They are also vulnerable to various environmental hazards, such as habitat destruction or climate change. Therefore, biological immortality does not confer complete protection against external threats. Organisms possess an internal biological clock, regulating various physiological processes. This clock dictates circadian rhythms, influencing sleep-wake cycles, hormone release, and metabolism. The capacity to reset this biological clock is a unique biological phenomenon. This reset mechanism allows an organism to adapt to environmental changes or disruptions. Such a capability offers a stark contrast to the life cycles of most multicellular organisms. Many multicellular organisms exhibit fixed biological clocks with limited or no reset ability. Beyond human perception, many animals possess extraordinary sensory capabilities. These senses allow them to perceive aspects of reality invisible to humans. Such abilities extend beyond mere longevity, encompassing a wider range of stimuli. This highlights the diverse ways life forms interact with their environments. The mantis shrimp possesses a highly complex visual system. This system significantly distinguishes it from most other species. Its visual capabilities far exceed those of humans. Mantis shrimp eyes can detect a broader spectrum of light. They perceive ultraviolet, visible, and polarized light simultaneously. This advanced vision aids in hunting, navigation, and communication. Color vision in living organisms is facilitated by specialized photoreceptor cells. Humans typically possess 3 distinct types of color photoreceptors in their eyes. These 3 types allow for the perception of a broad spectrum of colors. In stark contrast, the mantis shrimp exhibits an extraordinary visual system. Mantis shrimp can have up to 16 distinct types of color photoreceptors, enabling complex color perception. Mantis shrimp possess an extraordinary visual system, unique in the animal kingdom. Their eyes contain an expanded range of photoreceptors, far exceeding human capabilities. This allows them to perceive ultraviolet light, which is invisible to the human eye. They can also detect various forms of polarized light, offering a complex visual understanding. This advanced vision aids in hunting, navigation, and communication within their marine environment. The human visual system perceives a vast spectrum of colors and visual information. This perception includes nuances and variations that are often beyond linguistic description. Many specific color experiences lack direct, universally recognized names in human languages. This gap highlights the limitations of language in fully capturing sensory experiences. Consequently, some visual data remains unarticulated by existing human descriptors. The mantis shrimp is renowned for its advanced visual capabilities. Beyond vision, it possesses an exceptionally powerful and rapid predatory strike. This strike is one of the fastest known movements in the animal kingdom. It generates immense force, capable of breaking mollusk shells and aquarium glass. The mechanism involves a spring-loaded appendage that stores and rapidly releases energy. This biomechanical marvel allows for devastatingly effective hunting. Certain biological appendages, such as club-like structures, possess remarkable acceleration capabilities. These appendages can move at exceptionally high speeds through a liquid medium, like water. This rapid movement creates localized areas of extremely low pressure within the water. When the pressure drops below the liquid's vapor pressure, it causes the water to vaporize instantly. This rapid vaporization leads to the formation of small, transient, gas-filled cavities known as cavitation bubbles. The collapse of certain bubbles can generate significant energy. This rapid implosion creates a powerful shockwave. These shockwaves are capable of exerting immense force. Such force can be sufficient to break hard shells. It can even cause damage to robust materials like aquarium glass. The animal kingdom exhibits diverse and powerful movements. Some species possess striking forces disproportionately high relative to their size. These movements are among the fastest observed in nature. Such impact forces represent highly effective adaptations for survival. Regeneration is a remarkable biological phenomenon observed in certain organisms. It involves the capacity for extensive tissue and organ regrowth. Amphibians, such as salamanders and newts, are prime examples of this ability. They can regenerate complex structures like limbs, tails, and even parts of their brains. This process involves cellular reprogramming and coordinated growth. Studying amphibian regeneration provides insights into potential human regenerative therapies. The axolotl is a unique species of salamander, renowned for its extraordinary biological capabilities. This amphibian exhibits an unparalleled ability to fully regenerate lost or damaged body parts. It can regrow limbs, jaws, spinal cords, and even portions of its brain. This regenerative capacity extends to complex tissues and organs, often without scarring. The axolotl serves as a crucial model organism for regenerative medicine research. Axolotls possess an extraordinary ability to regenerate lost limbs. This regenerative capacity allows them to perfectly restore complex structures. The regenerated limbs are functionally complete, matching the original in every aspect. Remarkably, this process occurs without any scarring or fibrotic tissue formation. Their ability to regenerate limbs without impairment is a unique biological phenomenon. The regenerative capacity of certain organisms extends beyond simple tissue repair. This remarkable ability includes the regeneration of vital internal organs. For instance, portions of the heart can be fully restored after injury. Even complex structures like parts of the brain demonstrate regenerative potential. This advanced regeneration highlights sophisticated biological mechanisms for self-repair. Scar-free healing involves the regeneration of tissues that are structurally and functionally identical to the original. This process restores the tissue to its pre-injury state without any fibrous scar formation. Such complete regeneration is a rare characteristic in most vertebrate species. Typically, vertebrates heal wounds by forming scar tissue, which differs from the original tissue. The absence of scarring signifies a superior healing response, maintaining full tissue integrity. Neoteny is a biological phenomenon where an organism retains juvenile characteristics into adulthood. Axolotls exhibit a unique form of neoteny, distinguishing them from most other amphibians. They maintain their larval features, such as external gills and a caudal fin, throughout their entire life cycle. Unlike other amphibians, axolotls typically do not undergo complete metamorphosis into a terrestrial adult form. This retention of larval traits allows them to reproduce while still in their aquatic, juvenile state. Scientists are actively studying the axolotl, a unique amphibian known for its remarkable regenerative capabilities. The focus of this research is to understand the intricate biological mechanisms underlying their regeneration. Axolotls can regrow entire limbs, spinal cords, and even parts of their brains and hearts. This research aims to identify how these regenerative processes could be applied to human medicine. The ultimate goal is to develop new therapies for tissue repair and limb regeneration in humans.